My dear students, today's topic is what are the different digestive juices are secreted in the small intestine and what are their function. The question is, name different digestive enzymes juices in small intestine. You know, in my previous class it was described that gallbladder and liver plays a significant role in the secretion of biles. In the same way, pancreas also is involved to secrete the pancreatic juice. An inner lining of the small intestine is responsible to secrete intestinal juice or succus entericus. So what are the different juices that are secreted in the small intestine? First is the bile. Second is the pancreatic juice. Third is the enteric juice. Or this is called succus entericus. These three enzyme containing juice except bile are involved and they perform different digestive functions on different food compound and degrade or digest the small large complex macromolecules into smaller simplest form. First is the bile. What is bile? Bile is a greenish fluid that is secreted, formed by the liver and temporarily stored in the gallbladder. What is the situation? This is duodenum. This is the sphincter of the pyloric sphincter of the stomach, and the main part, this is the pancreas. This is the pancreatic duct, and this is liver. This is liver. In the liver, the upper surface of the liver, a muscular sac-like structure is present. This is called the gallbladder. Gallbladder contains a duct called the cystic duct, and two other ducts developed from the come from the liver, and they are called hepatic duct. This is the cystic duct, and two hepatic duct united to form a common bile duct. This is the hepatic duct. This is called the cystic duct or gallbladder duct. They together united to form CBD, common bile duct. This common bile duct united with the pancreatic duct. They united with the pancreatic duct. This duct is called pancreatic duct. Pancreatic duct. Through the pancreatic duct, pancreatic juice come and these poured through the duct into the duodenum. Through the CBD, bile is coming. The bile and the pancreatic juice mixed in this zone. This is called the hepatopancreatic duct. This is called hepatopancreatic duct. Common bile duct, hepatopancreatic duct, these two ducts united here and a sphincter is present. This is the sphincter of Oddi. Sphincter of Oddi. And then an ampulla is widened area. This is called the ampulla of Vater. Through this pore and sphincter and the passage, the two bile as well as the pancreatic juice enter into our duodenum, and they play a significant role in case of digestion of food. Then first topic is the bile. Generally, bile secretion in our body 700 ml to 800 ml, and mainly composition, their main composition is water. It's a liquid watery fluid. The water, obvious, the main component, majority part of the bile is water. Then it contains bilirubin and biliverdin. What is bilirubin and biliverdin? These are the degradatory product of the hemoglobin. When the red blood cell, the content of the red blood cell is degraded in the liver, the hemoglobin transformed into they produce two pigments, the bilirubin and the biliverdin. Then a other component is the cholic acid, cholic acid salt, sodium taurocholate and sodium plus sodium glycocholate. These two bile salt. Other than these two bile salt, other components are cholesterol. See, cholesterol and phospholipid. This is a lecithin. The major component of the bile, but here no enzyme is present. So how bile plays significant role in the case of digestion, particularly the lipid content, large complex lipid droplet are when entered into our duodenum. These large droplet fragmented into small piece by reducing their surface tension by the action of the salt, sodium taurocholate and the sodium glycocholate. That is the large lipid droplet. When the two salts of the bile salts acts upon it, the surface tension gradually decreases. Surface tension gradually decreases. As the surface tension gradually decreases, what happens? A large complex is split into several smaller fragments. Large fragment converted into smaller fragment by reducing the surface tension. This process is called emulsification. Emulsification is the process by which large fat droplets are fragmented into smaller ones. These are called micelles. This is a major digestive role of the bile. Bile also helps to absorb the fatty acid, glycerol and cholesterol in the lacteal. In that will be studied in our next class. Then bile. Bile generally reduces our lipid surface tension, and it transformed a large lipid transformed into smaller one, small producing many number of micelles, their surface area increase, so that the other enzymes of the pancreatic juice can interact in these lipid molecules, and they are fragmented into smaller and smaller fatty acid, glycerol, monoglycerol and diglyceride, and so on and so forth. Then this is the bile, main function of the bile in the case of digestion. Then the pancreatic juice. What is the composition of the pancreatic juice? Pancreatic juice is also a enzyme containing juice. Several enzymes are secreted through this pancreatic juice. In the pancreas, several acini present. These acini secrete these juice, and ultimately through the tubular structure, these acinal product enter into the main duct, and ultimately it is poured into the duodenum. What are the major enzymes present in the pancreatic juice? The second point is the pancreatic juice. Pancreatic juice generally contain most of the enzymes, amylolytic enzymes, second is the amylolytic enzymes that are called amylopsin. Amylopsin is a pancreatic carbohydrate digesting enzymes. Second is the trypsin, trypsinogen, chymotrypsinogen, carboxypeptidase. Trypsinogen, chymotrypsinogen, procarboxypeptidase. These are all the proteolytic enzymes. These are proteolytic proenzyme. These are the proteolytic proenzyme. Proteolytic. These are the carbohydrate splitting enzymes, the proteolytic enzymes, the protein splitting enzymes. And third is the lipase. This is called the steapsin. Another name is the steapsin. What are the major? The main question arises the same way when in the case of when we consider the gastric juice. In the gastric juice, the proenzyme is called pepsinogen. This pepsinogen is activated by the action of the HCl. But here, the trypsinogen is also inactive form. Chymotrypsinogen and procarboxypeptidase both are inactive form. But they are activated by another enzyme secreted by the inner lining of the intestine. This is called the enterokinase. Enterokinase. By the action of the enterokinase, this trypsinogen converted into active trypsin. The trypsinogen inactive form, this is zymogen, is a proenzyme, converted into active trypsin by the action of the enterokinase. When the trypsin is activated, this trypsin acts on chymotrypsinogen, by the action of trypsin transformed into chymotrypsin. These are the active form. These enzymes act on protein and degrade the protein into smaller form. So what are the product of these enzymes? First is the amylopsin. First product is the amylopsin. Amylopsin is a carbohydrate splitting enzyme that act on starch. By the action of amylopsin, a carbohydrate splitting enzyme present in the pancreatic juice, this converted into disaccharide maltose. The trypsin, second, the proteolytic enzyme, trypsin protein, peptide, peptone are converted by trypsin and they are produced dipeptide. Third enzyme, the trypsin or lipase, pancreatic lipase or trypsin. This pancreatic lipase, trypsin, acts on small micelle on fat, and this fat transformed into diglyceride and monoglyceride. Diglyceride and monoglyceride are formed by the action of the trypsin or pancreatic lipase. These are the main functions of the pancreatic juice. Then the third enzyme, and the pancreatics also secrete some amount of bicarbonate. This bicarbonate change the pH of the inner juice, and the pH level generally 8, slightly alkaline. In the slightly alkaline medium, these enzymes, particularly lipase, remain activated. So bicarbonate plays a significant role to maintain the enzyme activity of the lipase in the pancreatic juice. This bicarbonate also secreted present in the pancreatic juice. Third is the enteric juice. Third point, these juices are called succus entericus. Succus entericus. Succus entericus contain all type of enzymes, and these is the last enzymes, final step enzymes. They act on carbohydrate, protein, fat and nucleosides, and ultimately they are degraded all these complex organic food into simplest structure, that is the carbohydrate, complex converted into monosaccharide, peptide or dipeptide converted into amino acid, and monoglyceride, diglyceride, these are converted into fatty acid glycerol, and all other disaccharides present in our food also degraded, also digested by all the enzymes present in the succus entericus. Succus entericus mainly composed of mucus. This mucus is secreted by the goblet cell of the intestine, and the brush border cell, mucus and brush border cells of the succus entericus secrete different enzymes. Amylolytic enzymes in the group of amylolytic enzyme, maltase, but these got tage, lactase, sucrase, maltase, lactase, sucrase. Second is the proteolytic enzymes present in the succus entericus is the trypsin, and these are called dipeptides. Third group is the lipase. Fourth group is the nucleosidase and nucleotidase. What is the function of the maltase? Maltase act on, these enzymes act on maltose converted into glucose. Acts on maltose converted into glucose. Lactase act on lactose. Lactose is a sugar and ase is a enzyme. Ose is a sugar and produce glucose and galactose. Glucose plus galactose. Sucrose, it is also a disaccharide. Sucrase acts on sucrose. Sucrose producing glucose and fructose. In the same way, erapsin, the dipeptidase, acts on dipeptide and produce amino acids. Amino acids. Different amino acids are produced by the end product of digestion of the proteins. Lipase. Lipase acts on mono and diglyceride. Produce which substance that produced? Fatty acid and glycerols. These are the end product of the lipase activity in the succus entericus. And nucleosidase and nucleotidase, these acts on nucleotide and nucleoside. These nucleosidase acts on nucleoside and nucleotidase acts on nucleotide, and these, the product is base sugar, base plus sugar, base plus sugar. all the genetic materials, DNA and RNA, are degraded by nucleosidase and nucleotides in the case of succus entericus, that is the enteric juice. In the pancreatic juice, similar type of enzymes also present. This enzyme is called nuclease. Nuclease acts directly on DNA and RNA, and they produce nucleotides and nucleosides. These the end product of the nucleosides and nucleotides when reach the ileum, duodenum, and last end of the ileum and jejunum. The succus entericus acts on them and produce their structural unit, simple structural unit. These are the enzymes present in our alimentary canal.